Dr. Martha Delaney. She is a pathologist with the University of Illinois, their Zoo Path program. She's going to tell us a lot more about that. She has done a lot of really awesome stuff. Our background goes back, I don't know, I was like thinking 6 or 7 years ago. We did a little bit of work together at the Marine Mammal Center in California when she was doing a pathology rotation. So I'm super excited to see you again and have you on this program. So just want to hear a little bit more about what you do and some cool stuff or research projects that you're doing. So can you just tell us, I guess, a little bit more exactly of your role and where you work and what you do?
Sure. Great to see you, Lauren. Thanks. So I'm a pathologist, as Lauren mentioned, in the zoological pathology program. So I work every day as a veterinary pathologist, and I am specialized in that I work primarily with zoo and wildlife and aquatic species instead of dogs and cats and horses and cows and whatnot. So I did a training program at the Zoo Path program many years ago and then joined as faculty 2 years ago. And so from day to day, what we do is we provide diagnostic services for our several institutions here in Chicago. So the John G. Shedd Aquarium, the Lincoln Park Zoo, and the Brookfield Zoo. And so I don't know exact numbers, but through those 3 institutions alone, there's thousands and thousands of animals of varied species that we're responsible for helping the clinicians determine causes of death of some of these animals. And we also do clinical work in that if an animal has a lesion or a mass or something that the clinicians can remove surgically, and we will do biopsy work for them, meaning trying to figure out what that lesion is for them. So it's really great. We get to work closely with the clinicians, but our group, we function solely on the pathology side of things. So I will say everything from fish up to whales and everything in between. We do lots of birds and amphibians and reptiles, and then of course all the different types of mammals. So every day is fun. There's something new to look at, something new to learn. Yeah, so that's basically it on a day-to-day basis. Yeah.
Can you tell people watching this a little bit more? I mean, I think pathology is super cool, and just a little more like into more exactly what a pathologist does and like why it's important and what you're really looking for with an animal.
Yeah. So of course we don't want animals to die, but it's kind of a natural process. Animals die all the time at the zoo. Again, with thousands and thousands of animals, usually we'll have a necropsy. What the term in human medicine would be autopsy. We call it necropsy. And so basically it's a post-mortem examination. So we'll have animals submitted to us as carcasses, and then we'll do, we basically do a dissection. We open up their bodies and look at all the different organs, look for any, what we call gross lesions. So any evidence like abnormalities, maybe pneumonia or a mass in the liver or something wrong with the GI tract or something like that. And then as pathologists, what we'll do is we'll take samples from all of the major organ systems. So we basically take a sample of pretty much everything, brain and skin and all the organs. And then what we do is we fix them in something called formalin. And that's a pretty common fixative that's used across, human pathology, veterinary pathology. And after about 24 hours, the tissues are fixed to a point where we can trim them. We put them in these little cassettes, and then they go to a histology technician or histology lab, where the technicians will basically take our little pieces of tissue and they'll make, they'll cut very thin layers and put them on a slide, a microscope slide. And then they stain them and then they give us the slides. And then from there, we do a histologic or microscopic evaluation. So using a microscope, we'll look at all those different tissues and see if there's lesions or changes at the cellular level, right? So we basically take it from the animal's body, we look for gross lesions, and then we'll take it to the microscope and look for any microscopic lesions. And then together we write a full report, and hopefully we'll have some information to give to the clinicians or whoever submitted the animal. We're obviously looking for any kind of infectious diseases, neoplastic or cancer, and then any other types of problems that we can help with the management of other animals in the facility, or making sure that there's not an infectious disease going around, right? That could be a risk to other animals or even humans, what we would call a zoonotic disease. So we find our job, obviously I love pathology too, I'm a pathologist, but I think pathology is really, really important, especially now given our current circumstances. All of the people that are passing away from COVID infection, pathologists are looking at those people and trying to figure out what this virus is doing to their bodies, right? So besides the clinical information, the pathology is really important. So that's basically kind of in a nutshell what we do.
Well, that was great. My next question was like, I think people, the animals pass away and you don't hear much more beyond that.
Mm-hmm.
How
Mm-hmm.
pathology is such a powerful tool,
Mm-hmm.
not only for those animals that have died, or people or whatever it is, but how that can be used for the living animals, and
Yeah.
how what you do can really be related and transcribed into animals that are still in care, right? So that's awesome.
Yeah, and then being in the kind of the zoo world, there are a lot of species in the zoos that are endangered, right? And so we have populations in the zoos that they're being cared for to live out their lives. They're also very important genetically, right, to preserve their species going forward. So we want to know if any of these breeding animals might have some diseases that maybe they're not the best match for a certain animal if they have a predisposition to a disease. So we also are kind of surveying these animals, making sure that they're kind of the best genetic stock, so to speak, because in reality, a lot of these animals, they're only existing in zoos or in controlled situations, unfortunately. So we think it's really important for conservation of species as well. And then getting back to the zoonotic potential, one thing I like to tote is that it was a zoo pathologist that discovered West Nile in the United States, right? So she noticed a lot of crows were dying, and she started looking at them and realized, oh, we have this virus that was exotic, and now it's rampant in the U.S. now, right? So yeah, being part of that human-animal interface too. And I was taught by someone that mammals are mammals, right? And humans are kind of animals too, right? So thinking comparatively and looking across species, is something that what I love doing every day. So getting that case variety is fun.
Well, yeah, and that's another great point, is that a lot of your work and other work we've seen at the Marine Mammal Center, that things that people were looking at in animals, were also very relatable to humans, right? Human care, different illnesses that humans have, but they were seeing them kind of across species, which is really
that's another great point, is the interface there too. Is like, perfect example is the sea lions with the urogenital carcinomas, with the cancer, right? So using them almost as, obviously we care about the sea lions and we care about their health, and all, seeing like, well, why are these animals getting cancer in the wild, looking at ocean health, ecosystem health, and how that plays a role. But then also using the sea lions as a nice model, so to speak, for human cancers, right? So yeah, it's all comes together, and that's what's really cool about the field of pathology, is that it is kind of, you're talking about like translational, and it's always fun, and being part of different research groups, and yeah, helping kind of continue the knowledge, continue learning, increasing the knowledge, and yeah.
So I just have to ask this question because I'm just fascinated with what you do, but like what your day looks like, as far as, are you in the zoo, or is it just totally random?
Yeah, great question. So I should say that we have those 3 institutions that we will actually go to the institution and do the necropsies. We do a lot of what we call send-in work. So obviously, from the Marine Mammal Center, previously we would get cases from you guys, like you guys would do the necropsy and send us the fixed tissues, and we would do the histology report for you guys. We do that with a lot of other institutions. And then one thing I did fail to mention, one of our, we actually have 4 main clients, so to speak, here in Chicago. The 4th one, and I think is one of the most interesting and most fun, is actually the Cook County Wildlife Department. So Cook County, which is the county in which Chicago is in, believe it or not, has a ton of green space. They have a huge forest preserve. I think it's something like 70,000 acres of forested and like kind of like prairie land, right? So it's preserved for wildlife, and people go out and ride their bikes and run and whatever and ride their horses. But there's tons of wildlife too, with like coyotes and raccoons and skunks, and then obviously different kinds of birds and lots of deer. And so our job there is more of like a wildlife disease surveillance, right? So making sure, you know, is there a lot of distemper virus going around, or, you know, is there rabies going on in the bats, or, you know, things like that, or even some of the diseases that deer can have. So we kind of have our finger on the pulse for that. So with that, I would say any day we can get submissions from those places. And so we basically have a schedule. It's a little bit different now considering the circumstances. So that's why I'm working from home today. We try to have a few faculty in. Our offices are at the Brookfield Zoo, where we lease space. And then basically we have a group that is on duty for Brookfield Zoo, and then that wildlife, the Cook County Wildlife the carcasses will be dropped off there. And then we have a downtown team. So downtown is the Shed Aquarium and Lincoln Park Zoo. So basically we split it up that way. And if anything gets called in or submitted, we just, you know, we'll actually drive down to downtown to the aquarium or the zoo there to do the necropsies there. So we have a lot of times we have our little kits in our car. And then at Brookfield, we'll just go down to the necropsy room and take care of whatever's there. And then we have a residency training program. So basically, which is where I had done my training years ago. It's a 3-year program, which is typical for a veterinary residency program. And so we'll have residents that are on duty a lot, and then we're helping them every day with all their casework. So it's a lot of training in that regard. We do have veterinary externs. So these are veterinary students that are rotating through every couple weeks. You know, you've had them at the Marine Mammal Center previously. So working with them, and then obviously communicating with clients, which are the zoo veterinarians, right? So we learn, it's a two-way learning where we learn a lot from them, they learn from us, and, you know, about certain cases, and then overall like management of the zoo populations. So yeah, a lot of it just depends. It's all different every day, right? You know, like sometimes you have a lot of necropsies, sometimes you're a little bit light, but then obviously, as cases go, we're going to get slides back from histology lab. And so we call it double scoping. So faculty have microscopes that have 2 oculars. So we always sit every slide that we get with, you know, we'll look at with the residents, right? So any case they have, we'll double scope with them. Mm-hmm. So a lot of training, a lot of reading out cases, a lot of editing reports, so there's a lot of paperwork, a lot of writing. So that's the one thing about pathology. I love it, but I literally am writing reports like all day long, right? So that's basically it. I mean, you know, like I mentioned before, as pathologists, we never want anything to die, but things do. Yeah. And so, you know, you've done a lot of large animal necropsies. You know the logistics involved and the time it can take, and it's not just the necropsy, right? It's the cleanup and the disposing of the body and everything. So. You know, when we have the larger necropsies, we're usually kind of all hands on deck and just trying to get it done in a timely manner, in a safe manner. But yeah, a lot of reading slides, writing reports, and training. I guess that's probably the bulk of what I do every day.
and then a glamorous necropsy once in a while.
Yeah, that's my favorite part, I'll tell you.
I know. Oh, that's funny. Because me too. I mean, I loved it. And that's going to be my next question is just like, you've probably necropsied some crazy animals. I would guess, like huge animals, right, at the zoo. And like, you know, always sad that they've passed away, especially these animals that are there long term. But yeah, like anything that really sticks out is just like an amazing case that you've worked on.
You know, it's funny. I think that, you know, and this is probably really morbid and probably only what pathologists do, but I do have like a bucket list of animals that I want to necropsy at some point in my career. I've actually hit a lot of them. So obviously we're working with you and with Stranding Networks, doing a lot of, you know, marine mammal necropsies, which are cool, like the pinnipeds, the seals and sea lions, and then also the cetaceans. I love large whale necropsies, of course. One thing too, I think that is good to mention is that again, logistics of large animal, large whale necropsies. And of course, the most important thing for pathology is a preserved specimen, right? So when things start to rot, it gets harder and harder to figure out what's going on. That's one thing doing the wildlife where you kind of learn how to read through the rot, we call it. But sometimes we just have to shrug our shoulders and say, listen, I don't know, because the tissues are so autolyzed, which is rotten, right? So, but yeah, I guess in recent years, so the big ones are I should say, are fun, but they're a lot of work. And sometimes pathology-wise, they don't have the coolest lesions, but just doing them is cool. So I would say like elephants, hippos, I guess rhinos. Those are, one thing, it's different from whales. I mean, they're big, but their skin is so tough. So literally the knives, keeping them sharp is a challenge. And then just keeping up the stamina and getting it done, right? Admittedly, I don't like to do necropsies on great apes because they're so much like us, and I'm grossed out by humans. But they're very cool animals, of course, and then with them, we wear a whole other layers of PPE to protect ourselves from their potential viruses. Like, so a lot of, we call them old world primates that are from Asia and Africa are, a lot of them have SIV, which is very much like HIV. So things like that, we're trying to protect ourselves from. And then I guess, I really like doing, to be honest with you, pathology wise, I'm really getting into the amphibians and reptiles and of course fish. I mean, I think that they just have microscopically, and I guess, grossly, their organs are a little different. They're just a little different, right? And so it's to me like kind of relearning that stuff and getting more used to looking at lots of frogs and toads and snakes and lizards. They're kind of fun, and they have some cool lesions. So yeah, I guess I'm not. I love my marine mammals, so I'm not. But I love it all. And I guess for like a super interesting case. I don't know if I should share it, but no, but yeah, just the ones that, of course, the joke is too, if pathologists think something's really cool, it's usually really bad for the animal, right? So, we get really excited about certain infectious diseases because they have, you can either see the organisms or see the virus or something like that, the viral inclusions, but usually it's a bad thing for the animal or the zoo or the group. So, yeah. Trying to kind of not be too excited about the bad stuff.
Yeah, exactly. So can you talk a little bit about like just some of your background and training? We probably should have started there, but you've done a ton of schooling and like how you got to this point and why you wanted to do, I guess, go from, just being a clinician to pathology.
Sure. So I guess I'll start, as far as I can remember, I've always wanted to be a veterinarian. And then I think when you say the word veterinarian, most people think of the general practitioner working at a vet office that you take your dog or cat to, that they do vaccines and they'll do spays and neuters and they'll do basic sick visits. And usually that's kind of all you need. You get your heartworm medication, you get your deworming and, things like that. And like nail trims and the basics, which is so incredibly important. Don't get me wrong. I learned pretty early I didn't want to do that. I did an undergrad degree in animal science at UMass Amherst, and I did the pre-vet track, so made sure I get all the courses I needed. And of course, everyone says vet school is competitive, and it is, but I have to tell people if you want it, go for it, keep working. So I got into vet school at Tufts University in Massachusetts. And I'll say that when I was, I think in high school and college, I always volunteered at like the aquariums and stuff like that, and just like scrubbing tanks. And I volunteered at like wildlife places, feeding baby birds and stuff, and a lot of cleaning. And then I learned that, a lot of animals, especially in rehab, a lot of them died, right? And I was like, well, why did they die? And what happened? Like, did we do something wrong or that kind of thing, right? And a lot of times they're just going to die anyway, but so I did start doing necropsies kind of early on, even before vet school. And I was like, I really enjoyed it. I knew that. So then when I went to vet school, my first year, I started going to the pathology rounds every week. So seeing what went through the necropsy floor, and they would show lesions, and just, I realized, like you had mentioned, pathology is such a huge part of clinical medicine, right? And I think when people realize that, it makes learning all of the diseases so much easier when you think about it from that point of view. And that's when I realized, like, I really want to figure out why, what what these diseases are all about and why these things happen, why the tissues look like that, right? So I think early on in my veterinary career, I knew when I wanted to do pathology. And so I just kind of, did the Path Club, and I also did the wildlife clubs and stuff like that. I knew I liked different types of species. I love dogs and I love cats, but I knew I liked all the other stuff too. So yeah, I got through vet school and then I knew I wanted to do at least one clinical year before doing a pathology residency. Okay. And I'll tell you, I asked every pathologist I knew, I'm like, did you go straight to your residency program or did you do some clinical work or whatever? So I did an internship in large animal medicine at Washington State University. And the reason why I did that was because I felt like I had learned a lot at Tufts, who's very strong in small animal medicine, and they had all the specialties like surgery and oncology and radiology. I mean, they were very strong small animal, and they had a great large animal hospital, but it was mostly horses, and we didn't do a whole lot of like ruminant, like cattle and goats and sheep. Mm-hmm. So this internship gave me that year of like basically ruminant, like the hoof stock training in diseases. And I think I was a pretty good doctor, but a lot of my animals did go to necropsy, and I would always go down to necropsy and look at the lesions because I realized how hard it is to diagnose stuff as a clinician. Like you can do radiographs and you could do, other imaging like ultrasound and blood work, and animals are very good at hiding their diseases in a lot of cases. So I realized how hard it is to be a clinician, right? And I appreciated that. I still appreciate that day to day. And I always joke that we have the best diagnostic test, right? We can open up the animal and look what's wrong with it. Yeah. So from there, and I had found the DuPath program. I had met with some of the faculty at different meetings and talked to them, and I was like, I really, really want to pursue pathology residency, and specifically in zoo and wildlife species. So yeah, I got the residency, and then got the training there, or actually here, and then I decided to be an academic longer, and I did a PhD at University of Washington in Seattle at a medical school, which was interesting. That's a whole other story, but and I guess it comes down to anybody listening, if they're interested, if they're veterinarians and they want to do pathology, you can do pathology residency with clinical work or not before. And then there's the big question for our residents, like, do they do the PhD or not? Because nowadays, if you want to be in academia, the PhD is not a requirement, but it's always a bonus. And I'll tell you, like, it was a tough deal, but I learned a lot during my PhD, not just like the science, but just like managing, a research project and everything. So yeah, I'm really happy to be back in like the diagnostic world. We do little research projects. I like to be part of a team versus like the principal investigator of a research project. I've learned that from doing it, doing a PhD. So yeah, that's my advice to anybody listening if they're in this realm, that it's years of your life. To me, it was worth it. And I got the job I wanted, and now I'm really happy with where I'm at. Yeah.
That's awesome. I'm envious, a little envious. A lot of school, but your job sounds really amazing.
Yeah.
I know you've done some really cool stuff too, lots of cool field work. And are you working on any projects now?
Yeah, I'm trying. Excuse me. I like to diversify, right? So, and I was lucky enough. So when I lived in Seattle, even though I was working with like mice in the lab and wasn't like my happy place, I was lucky enough to kind of keep in touch with some of the marine mammal people. And because I was a quick flight to Alaska, I was able to go up there and do a couple. Did some belugas and some killer whales, which was awesome. And that was really, I love the field necropsies, and but admittedly, it's hard to make like a career out of just that, right? With government funding and stuff like that. And then just the flexibility of your day-to-day work schedule, right? So I'm really thrilled to do that kind of thing. I haven't been able to do it much lately, but in terms of research projects, I guess more recently, of course, with the COVID-19 pandemic and the SARS coronavirus 2, that's circulating in human populations. The big question is what animal did it come from? Because I think good assumption is it came from an animal host, that it was just there for who knows how long, and then there was spillover, so to speak, in the human population. And I'm sure, I think more recently there was some work I was involved with the tigers and lions that were infected at the Bronx Zoo. So I guess the zoo pathology world is quite small. So we were able to help out with that workup done by the pathologists there at the Bronx Zoo. So specifically what me and my boss, Karen Terrio, did was we worked on basically detecting the virus, the coronavirus, in the tiger samples. So none of these animals died, by the way. So this is an example of like ante-mortem pathology, so clinical pathology. So they were able to get some fluid from the lungs and the trachea, which where the virus happened to be, right? That's how the animal was infected, the respiratory tract. And they sent us those samples, and then we were able to use this technology. It's really called in situ hybridization. So basically using probe, so to speak, to bind to the virus in the sample and visualize it. So that's the part that we did. And that's something that we have established in our lab, mainly because we work with so many different species. There aren't like the tests and reagents available, that they are for dog and cat or human or mouse, for research and stuff like that. So that's something that we're hoping to kind of keep moving forward with that and developing new probes to detect other things, not just viruses and pathogens, but even different like genes or markers and tumors for these kind of more exotic species. So that's called RNAscope is the name of the brand name, so to speak, of this test or assay. So working on that. And then I'm always working with the residents. The saying is publish or perish, right? So we're always trying to publish, not just for our own selves, but there's not a whole lot of information out there comparatively for like wildlife, wild animals and zoo animals and aquatic animals in terms of pathology goes. So we're trying to always put out good information, a lot of retrospective studies, meaning looking back at all the archives of our animals and like, what do these frogs get? Or like or is there an outbreak of this disease in these animals, right? So we're constantly kind of cycling through and getting stuff published, things that kind of pop up that we think is worthwhile getting out there. One of my favorite animals are naked mole rats. So I've done a little bit of work with them. And it's funny because that's a great example of the intersection between like veterinary pathology and then human, and I guess I would say like lab animal pathology where there's pathologists working with scientists that study human diseases using like mice and rats and whatnot. So when I was at University of Washington, I worked with the pathologist there that did a lot of the modeling stuff, right? So one of my now mentors, Piper Truding, who's a very well-known mouse pathologist. She's like, well, what do you think about these naked mole rats? And I think of them as a zoo animal, right? And I was like, well, we have them at the zoos and they die once in a while. And, sometimes they fight and hurt each other and then they have babies and sometimes they take care of their babies and sometimes they don't. And it's just kind of like, they're just the naked mole rats at the zoo, right? Well, now they're a really valuable animal model because they rarely get cancer. They live a long, long time compared to other rodents like mice and rats. They live up to like 30 something years, which normal mice and rats live to like 2 years, right? So there's a lot of energy and excitement about them as lab animal, as animal models. And so I've been working now looking at our zoo populations, looking at what are common lesions, like what do we normally see in them. So we've had a couple papers with that. And then now I actually just finished writing a book chapter for a naked mole rat book for researchers who are using them. And that's been fun because kind of getting into more of the world of the naked mole rats that are used in like research facilities, right? And the differences there. So yeah, kind of dabbling in a lot of different things, but yeah, that's basically it. I think that's what else is going on.
That's about it. Your life sounds totally busy and really fun. That's awesome. That's really exciting to hear all that stuff. Well, that's all the questions that I have.
Okay.
Thank you so much for bringing this information to our group of followers. Obviously a little more, maybe marine mammal focused, but I think this is super exciting and interesting. You have such a fun job. So, and I'm excited to just reconnect with you.
Yeah. And
So
I miss the
yeah,
days we were doing necropsies together.
I know, exactly. That was super fun
Yeah.
days.
I should mention too that so the ZooPath program and Dr. Katie Colgrove, she's doing. I mean, I help her with the work too. But we still get a lot of samples from all the unusual mortality events. Either Southeast or Gulf, or we kind of get them from all over still.
Yeah.
Not all of the cases, of course, but we'll get a subset of them. So we do a lot of work for NOAA still. And of course, it just seems to be never ending, right? So,
Yeah,
so yeah, I think that to me, that's why I love the marine mammal work, is because they are sentinels of the oceans and the ocean health, and they're telling us something, right? So making sure we're listening to what they're telling us, right?
Yes. Yeah. And having people like you to be able to tell those stories is awesome. Well, thanks, Martha.
Thank you. So great to see you.
Yeah, absolutely. And we'll talk to you soon.
Yeah. Bye, everybody. Thank you.